will continue. Today we will continue with the questions from photosynthesis. Last in the previous. Oh, hey, can you hear me?
Yes,
Hello.
sir. Yes, sir.
You can hear me. But in the previous class, we have solved some of the questions. Okay, and can you remember those? Here is okay. I am starting. We are starting with the question. There is one question. Question number 9. Okay. Can you read it clearly? Hello. Hello. Can you read it? This is a question from some light reaction of photosynthesis. Okay. Light reaction. This is where does water splitting complex is associated with PS2 complex is situated. This is during the light reaction, the one thing that there is photolysis of water. Okay, photolysis that water is splitted. ये चले गल डाकी रे ऋषभ this water is splitted, okay, and this water splitting or the photolysis of the water, this is associated with PS2 system. So it is PS2 system, and this PS2 is located PS2 is located in the inner membrane of the thylakoid. Okay, granothylakoid. This is the granothylakoid. This one is the lumen, and this is the membrane. This is membrane. This is the width of the membrane, and this PS2 is located on the inner side of the membrane, and this water splitting complex also is কি হয়ে যাচ্ছে বারবার ক্র্যাশ হয়ে যাচ্ছে it is located at the inner surface of the membrane, so water splitting complex is also associated on the inner membrane of the inner side of the membrane of the thylakoid. So our answer is inner side of the thylakoid membrane. Okay, this will be the answer. And that is why when the water is splitted, that the hydrogen ion which is produced during this water splitting or photolysis, that hydrogen ion is deposited in the lumen of the thylakoid membrane. Okay, so this is the answer. Next question, we will go to the next question. Next question is, the membranous system of grana is responsible for trapping light energy but not ATP and NADPH2 formation, trapping light energy and also for the fixation of carbon dioxide for ATP and NADPH2 formation but not light trapping, for light capturing and also for NADPH2 and ATP formation. The product of the light reaction, the product of light reaction is ATP, NADPH2, H2, and it is oxygen. So these three are produced in the light reaction, and here the photosynthetic pigments, different chlorophyll and associated pigments are there which capture the light energy. So this for light capturing and also for NADPH2 and ATP formation. Our answer is this. Of course, along with this, they also produce oxygen. Okay. So, and this process, this production of ATP and NADPH2 associated with this oxygen is only carried out by non-cycling photophosphorylation. Okay, so this is also the product of the non-cyclic photophosphorylation. In the cyclic photophosphorylation, only in the cyclic photophosphorylation, only ATP is produced and no NADH2, NADPH2 and oxygens are produced. Next question. Next question. Which of the statement about the light reaction of photosynthesis is false? We have to find out the false statement. Okay, which one is false? First thing, here the PS1 and PS2 are located in the stroma of the chloroplast. PS1 and PS2 are located in the stroma of the chloroplast. This pigment system, okay, pigment system which is made of the light harvesting pigment, chlorophyll and other associated pigment, they are present in the membrane of grana. Okay, they are present in the membrane of the grana, that is in the thylakoid. All right. So here in this statement that PS1 and PS2 are located in the stroma, this is a wrong statement. And so our answer is this one. So this is our answer. Other statement that the PS1 and PS2 are linked by electron carrier. This is two pigment system PS1 and the PS2. These PS2 and PS2 are linked by the electron carrier. Different electron carrier are there. So electron from the PS2 move to the PS1 and then finally to the NADPH2. So electron carrier is there. So this statement is very correct. Chlorophyll have an absorption spectrum with pronounced peaks in red and blue light. This statement also is correct. Okay. The chlorophyll molecule, maximum absorption of the chlorophyll molecule is in the red and blue region of the spectrum. And that is why the photosynthesis also is more in the blue and red region of the spectrum. And maximum photosynthesis takes place in the red region. So this statement is correct. Chloroplasts have absorption spectrum pronounced peaks in red and light. Proton diffuse through protein channel, which are ATP synthetase molecule. This is also correct statement, because this thing I will explain in the next question while explaining you the chemiosmotic synthesis of ATP. That ATP synthesis, so this ATP synthetase molecule, this ATP synthetase molecules are located in the thylakoid membrane, and through that the proton, that is the hydrogen ion, they pass through. So our answer is A. This is wrong. And we know that because the light reaction, site of the light reaction is the grana thylakoid, and stroma is the site for the dark reaction. Okay, move to the next question. Which of the following statement is again incorrect about the cyclic photophosphorylation? Here, cyclic photophosphorylation. We know that there are two types of photophosphorylation. One is cyclic, another is non-cyclic. You know the phosphorylation. Phosphorylation is the process of production or synthesis, production of ATP. Okay, this is known as phosphorylation. ATP is synthesized from ADP and inorganic phosphate. Okay, this produce ATP. This production of ATP from the ADP and inorganic phosphate is known as phosphorylation. In photosynthesis, the phosphorylation process is light dependent. So it is called photophosphorylation. All right. It is photophosphorylation. And photophosphorylation takes place in two ways. One is cyclic, another one is non-cyclic. In the cyclic photophosphorylation, only PS1 is involved. But in non-cyclic photophosphorylation, PS, PS1 and PS2 both are involved. Okay, so this is the statement here. Our question is related to the cyclic photophosphorylation. Here it is the statement which are only PS1 is involved. So it is correct for the cyclic photophosphorylation. It occurs only light of wavelength beyond 680 nm is available for excitation. See, this PS1 in the photosystem, there is a reaction center. In the photosystem, there is a reaction center, and these reaction centers are made of a special chlorophyll A molecule. The PS1 has the chlorophyll molecule which is known as P700, that it absorbs light energy at or below 700 nm. Okay. This PS1 has P700, so at or below, so they absorb light at or below 700 nm. Okay, and PS2 absorb the reaction center of the PS2. This is actually known as P680. Okay, it means this one is made of the chlorophyll A molecule which has absorption maxima at 680 nm. So this absorbs light energy at or below 680. So when the light wavelength above 680 is available, in that case PS2 cannot work. PS2 PS2 cannot work when light above 680 nm is available. So at that time, only PS1 will work. And that is why PS1 work and it take part in the cyclic photophosphorylation. For the cyclic photophosphorylation, the PS1 alone is located in the stroma thylakoid. Okay. Stroma thylakoids are the thylakoids which connect the two grana. The membrane which connect two grana, they are called the stroma thylakoids. I think you the you can see this ওহো এটাকে আমি টেনে এনে ওদেরকে দেখাতে চাচ্ছিলাম ওটা হয়ে গেছে সিলেকশন হুম হুম see here this in this is the structure of the structure of a chloroplast, and within the chloroplast these are the membrane system. This is a membrane system. These are the grana. Okay, this is grana. This is grana. These are made of the thylakoid, and this thylakoid, this tube-like structure that is connecting, this is called the stroma thylakoid. Okay, these are the stroma thylakoid. This is a stroma thylakoid, and these are made of grana thylakoid. So the PS1 alone present in the stroma thylakoid, and in the grana thylakoid PS1 and PS2 both are located. So in the grana thylakoid the non-cyclic photophosphorylation takes place, and in the stroma thylakoid only the cyclic photophosphorylation can take place. Okay, next is only synthesis of ATP occurs. This is also correct in the this is only the synthesis of ATP takes place. This is, and synthesis of NADH occurs. This in the light reaction in cyclic photophosphorylation, this NADH2 synthesis does not take place. Okay, so our answer, we have to select the incorrect statement. So Here for the cyclic photophosphorylation, this is the incorrect statement that NADH2 does not occur. This is the answer. Okay, next come to the next question. This is the chemiosmotic synthesis of ATP. In chemiosmotic synthesis of ATP, proton diffuses through ATP synthetase from stroma into the thylakoid lumen, from thylakoid lumen into the stroma, from cytoplasm into stroma, from periplastidal space to stroma. Okay, this one, say chemiosmotic synthesis of ATP. This is the main theory for the synthesis of ATP. For ATP synthesis, chemiosmotic principle is the widely accepted hypothesis, and here during this synthesis of ATP, one is that the proton gradient is required. Okay, proton gradient is required. That is, at one side there will be very high amount of proton, and on the other side there will be no proton or very less amount of proton. Okay, so this a gradient is required, and during this chemiosmotic synthesis, the proton move through this membrane from the higher concentration to the lower concentration. In case of this photophosphorylation, in photosynthetic ATP synthesis, the hydrogen ion concentration is built in the lumen. ওইটার থেকেই রিলেটেড কোশ্চেন তো লাইট রিঅ্যাকশনের সঙ্গে এইসব see here it is I think now you can see here this is a diagram of the granathylakoids, just one thylakoid. So this this is the lumen. Oh. This is the lumen of the thylakoid membrane, and this one is the actually membrane of the thylakoid. During the light reaction, what happens? that hide in photolysis, the protons are produced. Protons are also transported from the stroma to the lumen. This is a stroma site and this is the lumen site. And during the electron transport in the mm, the- in the cyclic and non-cyclic photophosphorylation, protons are transported from the stroma to the lumen, and also protons are produced by the photolysis of water. So there is accumulation of hydrogen ion inside the lumen, and so a gradient is produced across this membrane of the thylakoid, across this membrane of the thylakoid. So the protons are transported from the lumen to the stroma, but it cannot do that. It will do through certain channel. These channels are provided by, this is the one that is called the, this is known as fundamental particle or F0, F1 particle. Okay, this is the F1, F0, F1 particle. This F0 portion, this is the F0 portion. Okay, F0, and this one is F1. F1 is protrude out of the membrane. This F0 has ATPase, ATPase function, or you can say ATP synthetase. ATP synthetase function. So all the proton that is produced within the lumen, they move across the thylakoid membrane from lumen to the stroma. From lumen to stroma and through F0, F1 particle. And during this process, that energy which is released, that energy is utilized for the synthesis of ATP from ADP and inorganic phosphate. So that this the hydrogen ion, they diffuse through ATP synthetase from thylakoid lumen to the, from the thylakoid lumen to the stroma. So this is the answer. So movement is from thylakoid to stroma. Okay, next question is a very easy one. That next question, photolysis of water result in the release of, photolysis of water that produces electron, proton, and oxygen. This photolysis of water produced when there is water molecule breaks, and this gives hydrogen ion, electron, and oxygen, and also water is produced. That is, the uh, this is a process that in photosynthesis we know water is utilized as well as water is produced. From the four molecules of water, when it is split, it produces four hydrogen ion, four electron, one molecule of oxygen, and two water molecules. That is, as much amount of or volume of water is consumed, half of it is produced. And that's why, so you, if you balance it, you can write this one as, that is 2H2O is 4 proton, 4 electron, and O2. So here our answer is this one, that electron, proton, and oxygen are produced. We will move to the next question. This is also related to the that already we have discussed, related to light reaction and the production of ATP. So the question is, thylakoid membrane bears several F0, F1 particle. Okay, several F0, F1 particle. This F0, F1 particle is also known as the ATPase or ATP synthetase. It is the synonym of F0, F1 particle. Which of the following is correct for the particles? Which of the following is correct for this F0, F1 particle? Statement 1, one of its parts F0 is embedded in the membrane and forms transmembrane channel that carries out facilitated diffusion of proton across the membrane. See, oh, okay. So this is the thylakoid membrane, and within this there is okay. This one is F1 and this is F0. All these are made of protein. Okay, this F0 portion, they are forming a channel. All right, through this, the proton that move. So this is correct that one of its part F0 is embedded in the membrane. F0 is embedded in the membrane and forms the transmembrane channel that carries out facilitated diffusion. You know, facilitated diffusion, diffusion may be simple, and in the facilitated diffusion, some proteins or the other substances help to move the substance across a membrane. So this F0 particle, F0 portion is helping to diffuse out the proton from lumen to stroma. This is correct. its other part F1 particle, its other part F1 particle protrudes out from the outer surface of the thylakoid membrane facing towards stroma. Yes, this one is exposed portion, not embedded in the membrane. This is exposed, and this is in the stroma side. Okay, this is the lumen. So F1 portion is in the stroma side, F2, F0 portion is embedded. So this statement also is correct. The third one, the catalytic site for ATP formations are located in the F1 part. Catalytic site, this is, that is the portion or the protein which catalyze the synthesis of ATP, that is enzymatic portion, that is the F1 particle. This F1 portion of the protein, they function as enzyme and it catalyze the synthesis of ATP. So this one is the ATP synthetase. So this is also correct. So all these three statements are correct. So our answer is all of these. So all the three statements that were given here, all the statements are correct. Okay. So next we will move to the next question. The enzyme ATPase couple the synthesis of ATP to the diffusion of proton. This is one we have already learned. So answer is this A. Come to the next. This is accessory pigments. Okay. Here, accessory pigment. Say, chlorophyll A is universal and this is the principal pigment. Because this chlorophyll A molecule carry out this light reaction. All right, chlorophyll A molecule. Photochemical reaction, reaction of light reaction is carried out by chlorophyll A. So this is the principal pigment. All right. This one is the principal pigment. And rest of the pigment, that is the chlorophyll B, then carotenoid. Right. Actually, carotenoids include carotene and xanthophyll. carotene and xanthophyll. But in the NCRT book, they have mentioned carotenoids and xanthophyll, but carotenoids actually includes the carotene and xanthophyll. Carotene and xanthophyll together is known as carotenoids. Anyway, this the carotene and xanthophyll, they are known as pigment. Accessory pigments, that is, it helps the chlorophyll A molecules. They absorb wavelength, light energy at a different wavelength, which the chlorophyll A cannot. So they can absorb more light, okay, from the different wavelength, and so help it. So this one is our answer, that accessory pigment allow plant to harvest visible light of wider range of wavelength. Wider length of wavelength. So, and this way they enhance the efficiency of photosynthesis. Okay, enhance the efficiency of photosynthesis. Okay. None. Hmm. Okay, next, say another question. Now come to some of the experimental portion of related to the photosynthesis. In the discovering, in the determining the different process that takes place, the different phenomenon takes place in the photosynthesis, there were several experiments carried out. And our next question is, question number 18 is, isotope popularly known to have been used in the study of photosynthesis. The isotopes, as you know, they are the elements having different mass. So the which are the elements that were used for the experiment, photosynthetic experiment. This I think before also in the previous class that I mentioned that for the evolution of oxygen in photosynthesis, it was a heavy isotope of oxygen was used. Oxygen, heavy oxygen, that one physiologist, Reuben, and his associate, they use water having heavy isotope of oxygen and prove that the oxygen which is released in photosynthesis that come from the water, not from carbon dioxide. Of course, this was initially it was uh, proposed by that Neil, okay, but experimentally it was proved by Reuben and his associate. Next is that is the carbon 14, the isotopes of carbon. It was for the dark reaction of photosynthesis. Dark reaction of photosynthesis where the carbon dioxide is reduced to carbohydrate, that was carried out by Melvin Kelvin, the biochemist Melvin Kelvin. Melvin Campbell, and he used that carbon dioxide, and in the carbon dioxide carbon was heavy. So radioisotopics carbon were used to determine the different intermediate product of the dark reaction, that in the process of carbon dioxide reduction. So our answer will be this one, that option B, where carbon 14 and oxygen 18. So these are the two heavy isotopes of elements that were used in the experiment of photosynthesis. Okay, next question is the this is the most abundantly distributed enzymes or protein of green tissue is. This is actually Rubisco. Rubisco, which is known as, that is RUBP carboxylase. This is the old name, but the new name is ribulose bisphosphate bisphosphate carboxy oxygenase or carboxylase oxygenase. You can write also carboxylase oxygenase. This is the most abundant protein in the world. On this earth, this is the most abundant protein. or the enzyme, because you know it is the photosynthesis that is supporting life on the earth. The By the process of photosynthesis, the food is prepared. The green plant prepare its own food, and that food is also for the all other heterotrophs. So entire life is depending on the photosynthesis, and this that's why this process has to be taken in a very large scale. And this rubisco is the enzyme which is for the synthesis of the food, that reduction of the carbon dioxide. That's why this amount also is very high. So this one is the enzyme most abundant enzyme on this earth. PP carboxylase, of course, the another enzyme which take part in the photosynthesis, but that is only for the C4 plant. Decarboxylase, that is the decarboxylation process, means a removal of carbon dioxide from a carbon compound. But this one, the carboxylase, it is for the addition of the carbon dioxide to a compound. So this is, so today I think today we will end the class here. And if you have any question, you can ask. Do you have any doubt? Or you can, in the next class also, you can ask it. Do you have? Hello? Yeah, Somnath. Somnath.